slide projector, and the first slide went up, and then the bulb went out. And for the next hour, I did an interpretive dance presenting my talk because no one could find the bulb. So hopefully this will go off with fewer hitches. But it's a real pleasure to be here. And my presentation today is going to be more or less a historical journey through expression analysis. We're going to try to really emphasize what I think are some of the important points about how we approach this overall problem. So since this is a continuing medical credit or education credit course, I have to provide disclosure. So I have titles and lots of titles. You've heard of Professor of Biostatistics and Computational Biology at Dana-Farber, Professor of Computational Biology and Bioinformatics at the Harvard School of Teach, Chan School of Public Health. I have many other academic titles. So being an academic, rather than paying me more, they give me more titles, which means I sit on more committees and waste more time. I'm on a bunch of advisory boards, none of which are relevant for the work I'm going to present here. I founded a precision medicine software company in 2012. That company provides software, so I disclose that on the official disclosure slide. But nothing that company does is going to be relevant to anything I present here, other than the fact that everybody deals with data, and data is really our currency for all of the analysis we like to be able to do. So I like to use quotes in my talks, and this is actually one of my favorite quotes. This quote really crystallizes the challenges and opportunities that are available with the data we can generate today. So every revolution in science, from the Copernican heliocentric model to the rise of statistical and quantum mechanics, from Darwin's theory of evolution and natural selection to the theory of the gene, has been driven by one and only one thing, and that's access to data. And if you think about that, that's really true. That when we think about how science advances, data is what allows us to test our theories and hypotheses, to either falsify them or validate them. And if we falsify them, data is the raw material that we use to develop new theories, new testable hypotheses. So you can see why I love this quote. In fact, the reason why I really love it is I made it up. So this is my intellectual selfie, and then once I made this up, I realized it was too long, so here's the Twitter version, 115 characters, so you can tweet it and make me famous. But it's really true. Data drives innovation. And one of the real revolutions we've seen over the past 15 to 20 years has been a revolution in our ability to generate large-scale quantities of gene expression data. So how do we do that, and what is that data useful for? Well, I'm going to give you a little history of expression analysis to really start this, because even in the earliest methods that people use, there are really important lessons for understanding how we analyze gene expression data today. So the human genome was sequenced in 2000. These papers were published in 2001. The draft sequence was available. I'm sure you all know of the history of the battle between the public and private genome projects. Having worked in the field at the time, it was a very interesting time and place to be, full of interesting characters, some of whom you love and some of whom you hate. And I won't tell you which those are, but we can talk about that later if you like. But it was a very interesting time to be involved in genomic science, because there's this wealth of data, but these real pronouncements about what the value of the data was. What was most interesting was it gave us, the Genome Project gave us, a reasonable catalog of the set of human genes. And that was really transformative in the way in which we think about biological systems and how biological systems function. So I came into the field in the early 1990s with a fellowship from the then National Center for Human Genome Research, now National Human Genome Research Institute, to work in genomics. But my PhD and all the work I'd done previously was in theoretical physics. So I had to learn molecular biology from the ground up, and I realized, well, most people here understand this, that there's some people who may be here, maybe watching this on YouTube, who don't really understand molecular biology the way I hope I do now. But this is the way I really started thinking about biological systems, that encoded within that DNA, there are elements of DNA called genes. And if I asked everybody in this room what a gene was, I'd probably get 100 different answers, 100 different definitions. But sort of the working definition that people have been using for a long time is that a gene is a region of DNA that encodes a protein. And even in saying that, there's a lot of difference in interpretation about what exactly the protein encoding piece is and how expensive it is, and whether isoforms are the same thing or different things. But we'll just have a gene being the region that encodes a protein. That gene, in order to make that protein, is first transcribed to RNA, and the RNA is exported from the nucleus, taken up by the ribosomes, and translated into a protein. Right? So this is what people often call the central dogma of molecular biology, DNA to RNA to protein. And when we look at this problem, we look at this simple paradigm, what we see is really the core of the place where we ask questions about what these genes do and how they function in developing the phenotypes that we'd like to be able to observe and understand. So when we really think about what the proteins do, it's a little bit more complex, that those proteins fold up into a three-dimensional structure. And in fact, that three-dimensional structure is fundamentally important because that structure dictates their function. And the analogy I always like to give is that I can take a piece of metal and I can use it to make a hammer, or I can make a wrench. And if I take that same piece of metal and make a hammer or a wrench, while I might be able to use the wrench to pound in a nail, I can never use the hammer to loosen a bolt, right? So the structure actually matters, and that dictates the function, and part of that function is regulating, for some of the proteins called transcription factors, it's regulating the expression of other genes. So when I say expression, I'm really referring to how much RNA is being made. So we have this really interesting closed loop, and at the end of this presentation, I'm going to come back to this idea that this regulatory process is really important for understanding biological systems. So what the genome did was it gave us a parts list, a catalog of all the proteins that make up a human cell. But what we recognize is that different cells in the body perform different functions. And so all those cells are taking the same fundamental parts of this and doing something very different. Now, when I say it's very different, it really isn't. Okay, all the cells have to consume oxygen and sugars and produce carbon dioxide and energy, but these cells are performing different functions in the body. So a neuron is going to be expressing a core set of metabolic genes that are also being expressed in a muscle cell, but the neuron is going to be making neurotransmitters while the muscle cell isn't. And so what we see is that in each cell type, well, I'm just keep on hitting the wrong button. In each cell type, well, there are core functions that are carried out in common. There are a set of functions which are carried out differently. And those different functions are represented in different sets of genes that are expressed. In the same way, when we think about normal tissue and diseased tissue, or healthy tissue and diseased tissue, what we recognize is that one of the differences between those tissues is fundamentally the expression of different genes which change the way in which those cells act. So even before the genome was sequenced, people were developing techniques to ask, are there differences in the genes that are expressed? And one of the earliest techniques was the use of northern blots. A northern blot is a really interesting technology in which we take RNA from cells and we load that RNA into a gel. And we use electrophoresis to separate the RNA based on sizes in that gel. So the smaller fragments are at the bottom, the larger fragments are at the top. I can actually run a sizing ladder. I can use RNA or DNA with known fragments of known size. And I can use that to benchmark where different sizes are in the gel. And then what I can do is I can take that acrylamide or agarose gel, and I can extract the RNA fragments onto a membrane, and then I take that membrane, and I take specific gene fragments that I want to know about, and I hybridize those gene fragments as probes to the RNA, which is now bound to this membrane. And then I can visualize that in a number of different ways. So one typical way to do that is to use radioactively labeled probes and then to visualize them by looking at an X-ray film exposed to that radioactive membrane and developing it. And so this is an example of a northern blot. And one of the things you can do is look at the presence or absence of different genes, where what really distinguishes the different genes from each other, what allows us to detect them, is their specific sizes. And this is important because the size of the amplification product is really, on the filter, is really a filter on the data. It's what allows us to detect the presence or absence of different genes. But one of the important things I have to do to compare the levels is I actually have to scale the data. What I tend to measure when I look at a northern blot is the darkness of these bands, the intensity. And in order to compare them, I can look at this and say, well, this one is clearly brighter than that. But I can only really say it's brighter if I know I've loaded the same amount of material. If I loaded 10 times the amount of material here than here, well, it is brighter. It may not mean that on a per-cell basis there's more RNA here than here. So what people typically do is they load some kind of normalization standard. And so in this case, it's what we refer to as a housekeeping gene. Actin is a gene that we assume is expressed at near constant levels across all the cells. And if I look at this, what I can do is estimate the intensities here and then scale the data for each one of these up or down in such a way that I'm scaling the actin levels the same way to really facilitate that kind of comparison. So I can do a quantitative analysis based on the assumption that I'm loading the same amount of material into each cell, into each lane in this gel, and that this gene down here, actin, is expressed at constant levels, and I can scale that and all the other things in that lane to make effective comparisons. Okay? So it's an important lesson, and it's one that carries through to all the other methods that we use to analyze the gene expression data. So quantification requires normalization. And in this case, normalization is based on the assumption that actin is expressed at constant levels. After northern blots and the invention of PCR, people realized you could use polymerase chain reaction, or PCR, as a way of quantifying the levels of different genes based on the RNAs that are expressed. And so people developed quantitative RT-PCR, and now this is a widely used technique that's used to accurately assess gene expression levels based on the detection limit you have of seeing fluorescence from a minimum number of RNA molecules once they're amplified into double-stranded DNA. So for each gene that I want to look at, what I see is a curve in detected intensity. There's a detection threshold. And essentially what I look at is when this curve crosses the threshold, after how many cycles of PCR. If I have a lot of material, I'm going to get to the detection threshold earlier. So I detect it in a fewer number of cycles. If I have less RNA, I have to do more and more amplification until it's detected. And so by running fixed quantities of RNA for particular genes, I can actually get a titration curve where very low levels of RNA are detected very late, very high levels are detected very early, and by looking at the number of cycles after which I detect the presence or absence of a particular gene, I can in fact determine its approximate expression levels, its approximate representational levels. So I can look at this and many different copies. There are always things that are below threshold that I don't see, but by looking at the number of cycles, I can back calculate the approximate amount of RNA. And so this technique was very widely used. It's still very highly used. People still refer to it as a gold standard. Although, again, there are a lot of assumptions. One of the assumptions is what I use for this titration curve, and the other is that just like in northern blots, I have to include some control genes for each sample that I assume are going to be expressed at about the same levels in each sample. I have to include some kind of housekeeping gene control. And I use that to scale all of my measurements. So the filter here on detecting what the genes are that are being expressed are actually the specificity of the two PCR amplification primers that I use, Quantitation requires normalization, and this involves comparison to standard curves, as well as the use of a reference RNA. And that normalization, again, is based on a number of assumptions. In particular, that the gene that I select for my standard is expressed at the same level in all the different cells. Now, quantitative RT-PCR is often used in gene at a time, but in the late 1990s, there were a number of studies that really looked at large-scale gene expression using quantitative RT-PCR as a way of measuring large numbers of genes. So this is actually one of my favorite papers. It was published in 1998, and this study looked at rat brain and looked at the expression of 100 different genes over different stages of development in rat brain. And what they were able to do was perform a very interesting analysis. And this was really the first example that I can find in the literature of someone using what we refer to as a heat map to represent gene expression levels. So they have a number of different genes in a number of different classes, and they depict for each one of these genes the intensity of its expression level in each row. They express its expression level as a gradient of intensity. So the darker it is, the more highly expressed it is. And so what you can see are lots of different patterns. They also represented in this paper as waves of expression, and they grouped gene expression together in clusters, what they refer to as functional clusters or expression clusters, based on the patterns in which these genes were expressed. And that was really interesting because what it allowed them to do is to map what they refer to as different trajectories of these different classes through the developmental cycle in the rat brain. I thought this was really interesting because in this early paper with RT-PCR, it really captures a lot of the ideas that are even used today in the analysis of gene expression. So after RT-PCR, to sequence even 100 or estimated the expression of even 100 genes was extraordinarily difficult. So after this technology was developed and deployed, people recognized that there were probably better ways. So in the late 1990s, a new approach was developed to assay gene expression, and that approach was the DNA microarray. There are lots of different flavors of DNA microarrays that were created. Some of the earliest were developed by Pat Brown and his students and colleagues at Stanford, and those arrays shown here at the top were arrays that were made by mechanically spotting DNA fragments that were complementary to the RNA sequences that were interested in assaying. So DNA fragments from individual genes mechanically spotted onto a glass microscope slide. Other technologies, like those developed by Affymetrix and Agilent, synthesized probes de novo on the surface of the slide. So in the case of Affymetrix, they used photolithographic techniques like those developed for the semiconductor industry. For Agilent, the technology they used was one which was based on using inkjet printers and using those to deposit the chemicals to do de novo synthesis. In either case, what these array technologies allowed us to do was to represent the thousands of genes in the genome on a single substrate to perform expression analysis. Now, the implementation of these technologies was slightly different. For spotted arrays, what one typically did was isolate RNA from two different samples. So you can think about this as a treatment and control sample, or a treatment sample and a universal reference sample, an experimental sample and a universal reference sample. In each case, though, we take the two samples, we isolate RNA, we create a fluorescently labeled molecule. So a fluorescently labeled target molecule, often complementary DNA made to that RNA, but we make a labeled target molecule representing the different genes in those samples, and we co-hybridize them together to a single array. And so if you look at the panel at the top, what you can see is different colors. And the way these are typically represented is to use red and green. And so if I take my two samples and hybridize them together, the red samples and the green samples represent, the red spots and the green spots represent RNAs that were present in one sample or absent or present in the other sample, and at the various levels of expression. So you can see some spots are red, some spots are green, indicating presence in one or presence in the other, but most of the spots look sort of yellow, suggesting they're present in both. In either case, though, what one can do is measure the relative levels of expression in those two samples and produce a genes by expression level vector. And by looking at hundreds of samples or tens of samples, anyway, some number of samples, get a matrix of gene expression levels. Single color arrays, in some sense, were conceptually a little easier. What one would do is take RNA from a single sample, create labeled targets, and then hybridize those to an array. In the case of Affymetrix, there were multiple probes for each gene. So what one would do, as illustrated in the bottom panel, is take those expression levels for the individual genes, the individual probes, and average over them to get an estimate of the expression level for each gene. But again, the end result is a vector for that sample showing each gene and an estimate of its expression level. And then around this data, a whole suite of tools and analytical methods were developed to really facilitate understanding how those expression levels change between different states. While DNA microarrays were extraordinarily successful and sequencing technology became less expensive, what happened was we moved beyond assaying just genes that we could place down on an array to think about doing de novo sequencing of RNA. And so, in fact, almost every project that you'd undertake today wouldn't use arrays, although arrays are still fairly widely used. But any project you undertake today is very likely to use RNA sequencing, or what we refer to as RNA. So what we can do with RNA-seq is extract RNA from samples. We run a standard sequencing assay. So we take the RNA, we convert it to cDNA, we do DNA sequencing. And then again, there's a pipeline that we've developed to either take those sequence reads, align them to the genome, or align them to a library of gene sequences. And in doing this, what we are able to do is estimate expression levels for individual genes, or in fact to go even farther and estimate expression levels for individual transcript variants in different splicing variants, even fusion genes that occur in some diseases. So it gives us a much finer-grained window into what's happening. But again, at the end of the day, what we have coming out of these assays is a vector of genes and the relative expression levels. In the case of RNA-seq, it's individual counts. In the case of DNA arrays, it was fluorescent levels. In the case of quantitative RT-PCR, the raw data that comes out are delta C gene values. But at the end of the day, what all of these technologies do is give us the opportunity to measure gene expression levels at a very fine-grained level of detail. And armed with that, what we've been able to do is to begin to think not just about single experiments, but to think more completely about how we tackle the problem of understanding disease development and progression. So I and my colleagues, like many others, have started to look at this overall progression of disease and to think about how we can use these modern technologies, including RNA profiling technologies, to address the entire life cycle of disease. So if you think about disease, every one of us carries some genetic risk, and what we'd like to do is to be able to estimate those. So a lot of the technologies we use for those are really to look at genes and gene sequences and genetic variants. But beyond estimating risk, what we'd then like to do is do a better job of early detection of disease. And once we've detected the presence or absence of disease, we'd like to be able to stratify patients into different groups, recognizing the fact that diseases which manifest and appear to be very similar to each other may in fact be different at the molecular level. They may make the tumor more sensitive to one therapy than another, or make a person much more likely to progress rapidly, or make their tumor much more likely to metastasize. So we'd like to be able to look at that kind of data and information, often using expression data, and to use that as a way of staging the disease, but then more importantly, selecting the most appropriate treatment option that's likely to provide a good outcome and improve quality of life. Now, as we started to develop these assays, and in fact a lot of my career has been built around the use of data that's come from DNA arrays. But as you might imagine, with the rapid expansion of DNA sequencing technologies, a lot of our work has shifted to using techniques like RNA-seq or genome sequencing to really understand that molecular basis for disease. All right? So we have technologies, but now the question is, with those technologies, how do we actually use them in a way that will allow us to make measurements that are meaningful? And so that raises the whole question of experimental design. So let's take a look at that. When we think about experimental design, a lot of people and this has been a really interesting experience for me over the course of my career, they look at these high-throughput technologies like RNA-seq or DNA microarrays, and they'll show up in my office, and they'll say, well, look, I've generated a lot of data. Why don't you tell me what this data means? And invariably, my first question for them was, what was your experimental design? And honestly, the experiments that we can analyze are those for which the person has actually thought of the experimental design ahead of time. So although a lot of people take this for granted, it's actually one of the most fundamental elements of any analysis that anyone's going to do, which is to think carefully about the design and what questions you want to ask of the data, and then make sure that you have an appropriate design to address those questions. So when we think about designing the experiment, the first thing we have to think about is whether or not we actually have enough replicates and the appropriate number of assays that allow us to estimate the expression and the variance and do meaningful comparisons between groups. Having done that, the next thing we want to do is then collect the data, process and normalize it so we can make comparisons. Once that's done, the next step is to perform a meaningful statistical analysis of the data. Some of that is an exploratory analysis, our experimental hypothesis. How do these two different groups compare to each other, and what is the meaning of those differences? So at the end of the statistical analysis, typically what we have are a set of differentially expressed genes. And what these genes represent are those which have the biggest average difference between our experimental groups. So in the standard two-class comparison treated and control, we might perform a t-test after normalization to do our statistical analysis, and then what we get are a set of genes with the associated p-values showing how different they are and how confident we are in the differences between those two groups. The last step is looking at a biological interpretation for the genes that we get out. And ultimately, that's kind of where the rubber meets the road. We don't just want a gene list. We want some insight into what those genes tell us about the underlying disease. So at that point, typically what we do is we look for overrepresentation of different classes of genes, different functional classes, different pathways, and that allows us to understand what's driving the different phenotypes that we see. So we'll take a look at each one of those elements in this pathway in a bit more detail. Okay, but first, why design an experiment? This is a fundamentally important question. And the reason, as I alluded to earlier, is that the design of the experiment really influences everything that you do downstream. It influences the analysis, and it influences the accuracy of the analysis. So if you don't put time into that experimental design, you end up in trouble. I remember when I was first working at DNA microarrays, one of my colleagues walked into my office one day, and he said, "Oh, can you help me analyze data? I have 60 microarrays." And I sat with him, I said, "Well, that's great. You know, what was the experimental question you had?" And the answer was, "I had 60 microarrays." And so I said, "Well, you know, what were the different conditions?" And he told me he had 60 different conditions. And when I looked at that, I looked at him, and I said, "There's not too much we can do, because with 60 different conditions, not really any thought put into what conditions you were comparing."